Through Theme 1 and HBHL, we've been able to support the scientific endeavors of brand new faculty that have been hired to really work at the interface between computational neuroscience, machine learning, basic neuroscience, and clinical neuroscience. There's been a broad variety of projects that have been supported through Theme 1. These include looking at and examining single neuron level activity as a means of predicting the behavior of an organism, or using neuroimaging strategies to analyze the anatomy and function of New World non-human primates like the marmoset, and using that as a bridge between traditional rodent models and human and clinical neuroscience. We've also supported methodologies for examining, using machine learning to examine the cellular underpinnings of more sophisticated neuroimaging techniques to better understand what kind of signals we're actually pulling out from these techniques. And finally, using multi-omics types of strategies in machine learning and big data as a means of identifying single subjects who are most likely to be at risk for specific disorders, or to better understand treatment response within a disorder. And finally, wrapped around all this is Neurohub, which is an exciting platform that can help investigators house and manage the data, disseminate these analysis strategies that I mentioned before. And again, this has not just broad implications for our McGill neuroscientific community, but also a more global neuroscientific community and activities.
I cannot report on everything, but I really want to report on our multifaceted, multidisciplinary discovery platform entitled Biocomputational Models of Neurodegenerative Disease that is headed by Dr. Alain Daguerre. So I think they are using basically Parkinson's disease mutation, misfolded protein as a model, trying to understand how disease propagation and how these misfolded protein can actually affect the survival of specific neural population. But it will go much beyond that, because whatever defined in Parkinson's disease can be applied to other neurodegenerative disease. The platform is interesting because a lot of innovative idea grants have actually expanded from the platform, trying to basically increase the lifespans of the mini brain organoid, others using them to understand better cellular function into Parkinson's disease. And in terms of McGill Western grant collaboration, there has been quite good progress into developing an animal, a marmoset model, a primate model, marmoset model of Alzheimer's disease. And others have used as well in those McGill Western collaboration, miniscope to look at deep cells into the hippocampus, into neural activity at the single cell level, trying to understand the role of specific cell population in memory pathways. So I think there is a lot of technology and fascinating subject going on in Theme 2, and I hope you're as excited as I am. Thank you.
If anything, Theme 3 has been marked by its variety. It involves many people across all of McGill and connects to Western as well. The support has provoked, catalyzed work at every level, from the very fundamental questions about cellular mechanisms, rodent models, getting prepared to study this as well in the marmoset, which is a new approach here at McGill, to asking questions about clinical populations and how we can connect the fundamental to the clinical, to developing devices and apps to actually implement these ideas and test them in trials. I think the first phase has been valuable, especially in bringing the many people involved at McGill who are strong cognitive neuroscientists, systems neuroscientists, to bring them to think really hard about application and to connect them to clinicians and industry. And what we're going to see in the next phase, I think will be quite exciting, will be building on that hard-won knowledge, making use of the systems and pipelines that have been put in place through HBHL more generally to allow this kind of translation, and really beginning to move from the bench to the bedside.
So if we start with the major discovery funds that proposed 4 distinct projects. One was by Lawrence Kermeyer and looking to creating ethical space for First Nation-led neuroscience research. And lastly, the Montreal Antenatal Wellbeing Study that was led by Kieran O'Donnell, examining how to better identify pregnant women who are at risk of adverse mental health outcomes. Looking into the innovative IDs program, so again, several projects that have been funded that were part of Theme 4. And just to give one example, so a project that was led by Erika Moody, which looked at a personalized approach to depression care, and how using healthcare records could help discovering adaptive treatment strategies. And finally, from the knowledge mobilization program and the implementation science sub-program, several projects have been also proposed there, including one that I've led on implementing cognitive health intervention in real care settings for people with schizophrenia.
One of the strategic goals and the distinguishing feature of the HBHL program at McGill is the fostering of innovation, entrepreneurship, and commercialization of research results into tangible products and services to improve the health of Canadians. This is done by the Neurosphere group, which is a group of 4 people that include an in-house entrepreneur that are active in all of these areas, interacting closely with the Innovation and Partnerships Office at McGill. It has several programs. One includes money, which is basically done through the Neuro Partnership Program with local incubators, accelerators such as CQDM and MedTech. It has an Ignite and Accelerate program for early stage funding of research and subsequent linkage to industry. It also has a direct mentoring activity with the investigators to help them identify valuable projects and place them in context and in contact with the appropriate partners. And finally, it has an outreach program, which is basically to connect all of this activity to local partners, including local industry.